you ingested that chicken or paneer, it got into your buccal cavity as a giant protein, which means technically you can call it as polypeptide. To make it simple, you can think of a giant protein. So giant you can imagine Hulk, you can visualize that Hulk. Giant. I'm sure we don't have that big door where it will allow the giant to enter it. The ultimate destination of protein is to enter the cell, but to enter the cell, it's giant. It cannot enter. Cell with door until large nisle. It's very small, like a dot. Therefore, these giant protein or polypeptides, they have to undergo digestion. And who will do that? Enzymes in various parts of our alimentary canal, isn't it? But when you chew, what happens to the giant protein? It is broken down physically. When you chew, what will happen to the giant protein? It is broken down physically, or you can say mechanically, isn't it? There is no chemical changes to the protein. It's just being broken down physically. The large particles are being broken down into smaller particles. That's it, but not digested yet. Is it clear?
Yes.
And in our buccal cavity, there is no protein chemical digestion. Why is that? Because in our saliva, there is no protein digesting enzyme. You can just get down with the physical digestion. And we have already discussed the structure of a protein, isn't it? It comes as a 3D shape. Giant protein comes as a 3D shape, and it has got two terminal ends. One end is with the carboxyl group, COOH, and the other end is with the amino group, NH2. Is it clear?
Yes.
Now in order to digest the protein chemically, we have to unfold it. Unfold it and make it a long chain so that it's easier for enzyme to act on it and carry out digestion effectively. Isn't it? So to do that, what do we have to do? We have to swallow it. Isn't it? So what you do, you swallow with the help of saliva, coming all the way from the salivary gland, isn't it? You swallow with the help of saliva, it will turn it into food bolus. It will get into your esophagus, our esophagus, and we have a so-called amazing movement called peristalsis, right? Peristalsis, one end when it contracts, the other end will relax. And this contraction will propel the food forward. You think of the toothpaste. You imagine yourself squeezing the toothpaste. When you squeeze the toothpaste, the paste goes up, right? Yes. So this hand has to relax. The other hand has to contract. And literally what you're doing, when you press it, it's equal to contraction. Did you get that? Yes. And again, when the paste reaches here, again you squeeze it here. At the other end to relax. Because of the squeezing it, equal to contraction, it will push the paste forward. Similar to that. Peristalsis, when the other end contracts, the other end relaxes, and these contractions will propel all this forward. Peristalsis. How does it spell it? Peristalsis. Peristalsis. Now the food has entered the stomach. Take this region as a stomach. Is it clear? Now the two important cells we need to associate here is chief cell and parietal cell. So chief cell, chief enzyme, they are the chief guest. Take it this way. Enzymes, they are the chief guest. Without enzyme, no digestion is possible. So whenever you think of chief cell, think of enzymes. So whenever you think of chief cell, you can straight away think of plasminogen. Because you don't have to get mixed up between the HCl source and the plasminogen source. So chief cell, it's related to chief cell. Enzymes are being chief, the enzyme they act as chief guest. So definitely chief cell will secrete plasminogen, not the HCl. That's how you clear out the confusion. Did you get that?
Yes.
And chief cell is there's a synonym. What do we call it? We call it peptic cell. You can think of chencho pep, isn't it? As a mnemonic, you can think of chencho pep. C for chief, P for definitely peptic cells, no confusion. That's how you clear up the confusion between with the synonyms. Synonyms, when there are names, technical terms, more than one, you get confused. So to clear the synonyms, come up with the mnemonics. Is it clear? Yes. By default, parietal cell is going to be opposite cell. And parietal cell will secrete HCl, right? Now you see, we have the materials required to digest protein, isn't it? But the thing is, to digest protein, pepsinogen, they have to wake up. Right now, pepsinogen, they are asleep. They are all asleep, just like you people here. You people are asleep, isn't it? We have to wake up. In your case, who is going to wake you up? Definitely me. Me, of course. Of course, not politely though. Okay, I don't know how to be polite. Of course, through scolding, isn't it, Adu? Yes. But who is going to wake up pepsinogen? HCl, in a very polite way. HCl doesn't carry my personality. Very polite. Wake up, Lamalu. How are you? Wake up, my dearest pepsinogen. You need to wake up and act on the protein. You have to digest the protein which is there in the stomach. So, pepsinogen wakes up, gets converted into pepsin, active form. But the thing is, again, we need to do one more thing. The 3D protein, they are in 3D shape right now. We have to unfold it. What do we have to do? Unfold. We have to unfold it and we have to denature it. That's what we call denaturation. Unfolding the 3D shape and making it a chain. It's just like, imagine a ball of thread. When you uncoil the thread, okay? It comes as a long chain, isn't it? Yes. Similar to that, HCl will act on the 3D shaped protein, it will unfold it and it will denature it. Now it is easier for enzyme to act on it there. Did you get that?
Yes, sir.
So now pepsin, can you see the scissors here?
Yes, sir.
Do you really have scissors in your stomach?
No.
So what do you think it could be? Yes, it is representing, simulating. Yes, it is simulating pepsin, right? So pepsin, what they're going to do? They're going to cut off the chains of, long chains of proteins, which we call polypeptide. Peptide. Poly means many. Peptide refers to proteins. It's like this. It comes like this. Can you see the round structures here?
Yes.
They represent amino acid, okay? And can you see the bones here?
Yes.
So the bones between the amino acid, we call it peptide bones. To digest the protein, we have to break these bones, peptide bones. Who is going to break it? Right now, in this case, in our stomach, pepsin is there to break the bone. But pepsin cannot break each and every bone. They cannot do that. They cannot do that. They cannot break each and every bone. They can go on to break some bones, but they will still remain as a large peptides. Proteins will still remain as a large peptide. You can call it as proteoses, not proteases, okay? Proteases, it is ases. If it ends with the suffix ases, it will become enzyme. Proteoses, or you can say peptones. What you can call is peptones. Imagine you're doing a temporary job to earn some pocket money. Imagine you are crushing a rock. Go to Deche. Unless you are a Hulk, isn't it? So, Laluji, yeah, pieces of some part pieces, very small portion again. Some they remain as a large rock. Some remain as a very complex Laluji, yeah, large peptides. But stomach, mostly the proteins will remain as a large peptide. The next destination will be the duodenum. And you know what's duodenum, isn't it? It's the first part of small intestine. But before that, before we move on to duodenum, I would like to recall the two sphincters, okay? Very two sphincters, what are they? Large sphincter or lower esophageal sphincter. And we have here pyloric sphincter. While the stomach is busy digesting the food, remember they don't digest silently. They have to make noise, no, they have to move a lot. Stomach is always churning, there are a lot of motion, movement. So these two sphincters must be closed tightly to prevent the acid reflux and to prevent the entry of pre-digested food into the duodenum. You got that right?
Yes, ma'am.
Sometimes when you take a large meal, it puts a pressure on LES, and when LES gets weakened, the food regurgitates back, and you know that the stomach, food coming from the stomach is highly acidic. Remember the pH is around 2, highly acidic, and when it goes back, when it flows back, regurgitates into the esophagus. Our esophagus doesn't have a thick mucus layer. That's when it causes inflammation and causes heartburn, the symptoms of GRD. If it persistently occurs, it is definitely GRD, isn't it?
Yes.
Okay. But why do you think our stomach doesn't get burned by the acid, by our own acid? Yes, we have got thick mucus layer, thanks to the goblet cells, isn't it? Mucus cells, the goblet cells over. Okay?
Yes, ma'am.
Then another question I have for you, okay? Why protein digesting enzyme, in particular context of the stomach, in this particular context, you can see the protein digesting enzyme is no other than pepsinogen in us, in us. Baby na emerging program to karo. So manchik na emerging pepsinogen, why it is secreted as inactive form? That's the question. I have to form the secretary watching. Manchik stomach was the already protein is lowa. Ani protein this digest bini putulo, ani protein bini denura. Inactive form the secret bishavilo. Now activate when they get the signal that they have a protein here and jump to activate semen. Azum chi there ilo ina. Raise your hand if you agree with her. Correct, agree with me? Yes, definitely. So mugila then more. To prevent self-digestion, it's so abstract, you have to detail that. If you need to score full marks, if you want to score full mark, make sure you get down with the detailing. Definitely correct. Move on now.
Yes, ma'am.
Now you see, the proteins and the large peptides, they are entering the duodenum. And remember, the food in the stomach, are they fully digested?
No.
They are pre-digested. Pre-digested, or you can say semi-digested. Semi-digested. Are they acidic or alkaline? So obvious, right? Obvious answer. Acidic food. Which lolo? Acidic food. So this type of food, acidic, highly acidic, pre-digested or semi-digested food, what do we call it? It's correct. We call it as chyme. Now the chyme, let's say, chyme is entering the duodenum. Chyme is entering the duodenum. So duodenum enter with it. But the thing is, duodenum nalu, we don't have much thick mucus layer unlike stomach. So we have to neutralize it. Acid neutralize the otuka. Neutralize corner. So we have to neutralize the focus on this portion. We have to neutralize the chyme. Which means we have to neutralize the acid that is coming along with the chyme. So how to neutralize the chyme? Neutralize as soon as the chyme enters the duodenum, if I make use of an analogy, as soon as there is a knock on the door, you get a signal that someone is standing at the door, and what you do, you go there and open the door, isn't it? Yes, as soon as the chyme enters the duodenum, innermost layer, the innermost layer of duodenum called mucosa, it gets a signal that, oh, the chyme has entered, and then the mucosa of the duodenum will release a hormone called cholecystokinin. I'll freeze it. Okay. So in short, you write it as CCK. CCK. change to pharmacy maybe alto ka bachimani cholesterol. Now CCK will enter the bloodstream. It's a hormone then. Hormone, they always enter the bloodstream. Hormone trouble be through muscle bloodstream na trouble go. Who dug this na trouble be so. So they will enter the bloodstream. Blood na le pete le gati jo pancreas na jo. Gallbladder na jo CCK the gallbladder le target pro pancreas se target pro. They were the tato na. Since we are dealing with protein digestion, we don't have to bother about gallbladder. So let's forget it for a while. Don't sleep. And thus, SL will not be there to wake you up. So, polycystokinin will target the pancreas and it will stimulate the pancreas to release what? Pancreatic juice. Pancreatic juice will come all the way from the pancreatic duct and the hepatopancreatic duct and then it will enter the duodenum. And what are the enzymes, protein digesting enzymes in the pancreatic juice? Trypsinogen, trypsinogen, and co-carboxylpeptidase. Now you see, along with the enzyme, there is going to be bicarbonate salts as well. So bicarbonate salts and these are very important. And these bicarbonate salts will neutralize the what? Neutralize the acids. And acid neutralized baby color, it will make it alkaline. And then that's how it creates a perfect pH for these three enzymes to function in the duodenum. Around let's say pH around 7 to 8 bishaki. pH is the best function unlike the stomach, which is at pH 2, isn't it? Now trypsinogen is such a big deal of enzyme here. There's some potential in trypsinogen, but we potential unlock being the loop, we have to activate them. Right now they are all asleep. Why do you think they are asleep? Why do you think they are secreted as proenzymes? To prevent the digestion of pancreas. Pancreas, remember, they are made up of proteins. If they are secreted as active form, they will start digesting the proteins which are there in the pancreas. So pancreas is damaged without them. They are secreted as inactive form. Proenzymes or zymogen cell. Now you see what will happen. So trypsinogen, to activate the trypsinogen, to unlock the potential of trypsinogen, we have an enterokinase all the way from a cell called enterocyte. And enterokinase will fall under the category of brush border enzyme. Why brush border enzyme? Because you see, enterocyte, they are beneath the villi, and whatever enzyme secreted by enterocyte, okay, these enzymes are right at the top here, and these villi look like a brush. So since they are sitting right here in the brush-like structure, they are known as brush border enzyme. Did you get that? Yes. So enterocyte will go on to activate trypsinogen into trypsin. And now what do you think trypsin will do? Trypsin will, first of all, for a priority mission, it will digest proteins into correct large peptides. Then trypsin will go on to activate these two other friends. Who are they? Carbotrypsinogen and co-carboxylpeptidase. Carbotrypsinogen will be activated by the trypsin into chymotrypsin, and same way now chymotrypsin will digest proteins into large peptides. And the active form of carboxylpeptidase will digest the large peptides into dipeptides, and some they will be digested into amino acids. And dipeptide and remember the protein digestion completely says some they will still remain as large peptides anyway. Complex okay? Okay, alright. Now what's going on with the proteins, the large peptides and the dipeptides, amino acids, which will it will enter the ileum. Take this as an ileum. Take this portion as a ileum. And if you see our intestine linings are lined with villi. Villi purpose is to increase the surface area for absorption. Okay? You should know that beneath the villi, there is a cell called enterocyte. There's a cell called enterocyte, or you can say intestinal epithelial cells. You can say intestinal epithelial cells. Now you see, these proteins, you can see some are converted into amino acids already. But some they are still as a large peptides. Some they are still as a dipeptides. They are still as a dipeptides. Now the next mission is that they are in the lumen of the ileum, lumen lung cavity. Takes as a ileum. Nampana do the ileum nampana amino acids to large peptides, dipeptides system. Now what they want to do is they want to enter the enterocyte over there. What do they want to do? They want to enter the enterocyte. But before that, we have a brush border enzymes, dipeptidases and aminopeptidases. What they will do is they will make sure that they are digested into amino acid. Dipeptidases will cut the bone cut baby. Okay, it will convert it to amino acids. Same, aminopeptidases will do the same. And now, what's the mission? They want to enter the epithelial cells or enterocyte. They want to enter it. Now to enter, they need to undergo secretory clearance phenomena. What they need to undergo? Secretory clearance. And what is the secretory clearance? You see, we have these channels here, SGLT, sodium glucose transporter. What do they have? What do we have here? Sodium glucose transporter. We have sodium glucose transporter. We have H plus channel as well. We have H plus channel. But amino acid they can't enter alone, remember? Those amino acids they can't enter alone. To enter, they have to come with the ID card. And what is the ID card? Underlying budget, the ID card is sodium. Remember when we take food, the food contains sodium, HCl, this will be right? NaCl, this will be the food contains sodium. So to enter, for amino acid to enter, they have to come along with the sodium ions. And you can see two substances entering at the same time in the same direction. This type of transport we call it co-transport. Co-transport. Is it clear? Yes. And no usage of energy at all. No usage of ATP. ATP used because they are moving from higher concentration to lower concentration. Along the concentration gradient, not against, along, higher to lower. In this case, we don't have to make use of ATP, but we do need help from channels, remember? We do need help from the transporter, isn't it? So this type of mechanism of absorption, we call it facilitated diffusion. What do we call it? Facilitated diffusion. Why facilitated? Because these transporters are there to help them pass through. These transporters are there to help them pass through. Why diffusion? Because it fulfills the definition of diffusion, moving from high concentration to low concentration. So this tells us amino acid absorption is not necessarily the active transport, it's purely through facilitated diffusion and co-transport. Did you get that? And there are also an H plus channel that will allow amino acid to enter, but again along with the H plus. Along with the H plus. Can you see that? Facilitated diffusion. And the mechanism of transport is co-transport. Okay? Now you can see amino acid has entered the enterocyte. But what about this? What about this? And what about this? Anilium. These smaller peptides, okay? These smaller peptides or dipeptides, they can also, these channels can also allow them to enter, remember? And also, directly cell here engulf with the cell here engulf with the endocytosis long engulf with the right? So, now we have to digest them again, remember that. So, to be on a safer side, don't worry, we have peptidases there. What do we have? We have peptidases. Example? Dipeptidases. Dipeptidases. We have dipeptidases that will again digest this and convert it into amino acids. However,
Yes.
did you get this? And again, now we are at the borderline, borderline between the bloodstream and the enterocyte. We have a channel here. What do you have? We have a channel that will allow amino acids to enter along the concentration gradient. Along the concentration gradient, doesn't require any ATP, so it's purely facilitated diffusion. Oh, the absorption of amino acids purely facilitated diffusion. Then what about this? It is sodium, potassium to maintain the gradient. To maintain the gradient. And by maintaining the gradient, we achieve electrolyte balance. We achieve what? Electrolyte balance. Did you get that? Yeah, amino acid blood blood. Now after entering the blood, what will happen next? It's obvious, you can make it up from the flow chart. After entering the bloodstream, it will transport it to the liver. It doesn't look like a liver. Liver should come in this shape, isn't it? It will transport it to the liver, and liver will detoxify it. Liver will detoxify it, and then it will send it to our body cells. Now the amino acids it will finally enter the body cell. And you see, once it enters the cell will be the use benigutu. What's benigutu? Use benigutu. Cell will use it for repair. Cell will use it for growth. You were born as an infant, and you see, you have grown a lot, isn't it, Alu?
Yes,
in terms of brain wise, in terms of body size, in terms of height, you have grown a lot. And how did you grow? By using all these proteins, the amino acids. Okay? If some cell organelles are damaged, you can also use those amino acids to synthesize new cell organelles. If some structural proteins are damaged, you can also synthesize you can also use those amino acids to synthesize structural proteins. Let's say you don't have sufficient enzymes, you can also use the amino acid to synthesize some necessary enzymes that you don't have. You see, so what do we call this use, brother? We call it assimilation. What do we call it? Assimilation. So the first stage is digestion. Before digestion, what do you do? Ingestion. Digestion. After digestion, when the digested substances enter the bloodstream, that's what we call absorption. And after it reaches the cell, what do we call? Assimilation. We use it for greater purpose. Amino acid emerging, what's the greater purpose for amino acid? For growth, for repair, to build the muscles. To build the muscles, isn't it?
Yes.
And to build the structural proteins. To build some cell organelles, to build some enzymes. So this is assimilation of proteins. And as a pancreas, they don't know when to release. So as a messenger, we have cholecystokinin. When to release, we have to signal it. And who is acting as a signal or messenger? Cholecystokinin, all the way from duodenum mucosa. Did you get that?
Yes,
So that is protein digestion, absorption, and assimilation. Did you get it?
Yes, sir.
I'll assign you with some tasks, okay? What you're going to do is, you're going to write a thank you letter to HCl. Write a thank you letter to HCl. Did you write thank you letter to pancreas?
Yes.
Now you're going to be specific, okay? Specific. Write a thank you letter to trypsin. And write a thank you letter to dipeptidases and aminopeptidases. Now next what you're going to do is, you're going to simulate SGLT, sodium glucose transporter, and H plus channel as a gatekeeper. Next, you're also going to thank enterokinase. You know why you should thank enterokinase, isn't it? Without enterokinase, I don't think protein digestion is possible in the duodenum. Is it possible?